The overall goal of this procedure is to demonstrate a simple and highly reproducible protocol for the generation of recombinant arenaviruses in FDA-approved Vero cells for potential vaccine or vaccine vector seed development. This is accomplished by first carefully preparing the plasmid DNA transfection mixture. The second step is to transfect Vero cells with the plasmid DNA transfection mixture. Next, it is necessary to scale up transfected cells into 100 mm dishes. Ultimately, the final step is to confirm a successful virus rescue by immunofluorescence using wild-type virus, or by fluorescence microscopy and Gaussian luciferase expression using trisegmented virus.
Compared to existing methods, such as rescues and murine cell lines, this protocol generates arenaviruses in Vero cells which are approved by the FDA for the development of vaccines.
In addition to the generation of potential vaccine seeds, the reported gene-expressing trisegmented virus can also be applied for the identification of antivirals against arenaviruses in high-throughput screens without the need of secondary assays for viral detection.
First, prepare 250 microliters of optimum media and, depending on the virus rescue, 10 to 12 micrograms of lipofectamine 2000 per transfection. Incubate for 5 to 10 minutes at room temperature. During this time, prepare the plasmid transfection mixture in a separate microcentrifuge tube using the recommended amounts for each virus rescue. Bring the final volume to 50 microliters with optimum. Next, add 250 microliters of the optimum lipofectamine mixture into the plasmid DNA transfection mixture and incubate for 20 minutes at room temperature. To prepare Vero cells, first equilibrate the PBS, DMEM, 10% FBS, 1% PS media, and the trypsin EDTA mixture to 37 degrees Celsius. To harvest the Vero cells, wash the cultures twice with 5 milliliters of PBS. Then add 1 milliliter of trypsin EDTA for cell detachment. If necessary, gently tap to fully detach the cells. Now resuspend the cells in 10 milliliters of complete DMEM in a 15 milliliter centrifuge tube. Then centrifuge the cells for 5 minutes at 1000 RPM. Resuspend the cells in 10 milliliters of complete DMEM. Count the cells and adjust concentration to 1 × 10⁶ cells per milliliter. After the 20 to 30 minutes room temperature incubation, add 1 milliliter of Vero cells to the optimum lipofectamine DNA plasmid mixture and incubate for 5 minutes at room temperature. Now, seed the lipofectamine DNA cell mix into 6 well plates, gently shake, and then place in the incubator. After 16 to 24 hours post-transfection, remove the transfection media and add 2 milliliters of infection media. Culture the cells for an additional 48 hours. Next, to passage the cells, remove the tissue culture supernatant, wash twice with 1x PBS, and trypsinize the cells as shown earlier. Then resuspend the cells in 1 milliliter of complete DMEM. To pellet the cells, centrifuge for 5 minutes at 5000 RPMs and 4 degrees Celsius. Carefully resuspend the pellets in 1 milliliter of infection media and transfer the cells to a 100 mm dish. With infectious media, bring up the total volume in the plate to 8 milliliters, a sufficient volume to prevent drying of the cells as well as for concentrating the virus. Gently shake the 100 mm dish for a uniform monolayer of transfected cells after culture. After 72 hours, collect the tissue culture supernatants, centrifuge to remove the cell debris, and store the supernatants at -80 degrees Celsius. The day before titration, wash the Vero cells twice with PBS and trypsinize. After resuspending the cells in 10 milliliters of complete DMEM, count the cells with a hemocytometer and adjust the concentration to 4 × 10⁴ cells per well. Then, prepare 96 well plates to reach 80 to 90% confluency the next day. Gently shake the plates by hand to get a uniformed distribution of the cells. Culture the cells for overnight. On the day of infection, check the cells under the microscope to confirm a monolayer before proceeding with the infection. Now, serially dilute the virus-containing tissue culture supernatants that were recovered earlier in Optimum in a 96 well plate. Then aspirate the media from the seeded Vero cells, wash twice with 50 microliters of PBS, and infect cells with 50 microliters of the serially diluted virus. Infect the cells for 1.5 hours at 37 degrees Celsius and 5% carbon dioxide. Next, remove the virus inoculum, add 100 microliters of infection media per well, and incubate the cells for 16 to 18 hours. Now remove the tissue culture supernatants from each well. Fix the cells with 4% formaldehyde diluted in PBS for 15 minutes at room temperature. Then remove the formaldehyde and permeabilize with 0.1% Triton X100 at room temperature for 10 minutes. Next, aspirate the permeabilization solution and wash 3 times with 100 microliters of PBS. Lock cells with 2.5% bovine albumin serum. Dilute the primary antibody specific to an arenavirus protein in 2.5% BSA and centrifuge for 15 minutes at 3500 RPM. Incubate the cells with the antibody for 1 hour at 37 degrees Celsius. After 3 PBS washes, add an appropriate fluorophore conjugated secondary antibody for 30 minutes at 37 degrees Celsius. Remove the secondary antibody and wash 3 times with 100 microliters of PBS. Next, titrate by counting fluorescent focus forming units using fluorescence microscopy. Similar to wild-type recombinant arenavirus, titrate the trisegmented virus by first serial diluting the virus present in the tissue culture supernatant. Then infect Vero cells in 96 well plates. After 16 to 18 hours of incubation, titrate by counting fluorescent focus forming units using fluorescence microscopy. Alternatively, measure successful virus rescue by Gaussian luciferase expression using 100 microliters of tissue culture supernatants in a 96 well white plate. Set up the luminometer. As per the manufacturer's recommendations, add 50 to 100 microliters of Gaussian assay solution to each sample. Then measure the Gaussian reporter gene expression with the luminometer. As a negative control, measure Gaussian expression from TCS of wild-type virus-infected cells. Arenaviruses are enveloped negative-sense RNA viruses with bisegmented genomes. Each segment uses an ambisense coding strategy to direct the synthesis of 2 viral proteins in opposite orientation. 2 types of rescue plasmids are used for the generation of recombinant arenavirus in Vero cells. PCAG's expression plasmids direct the synthesis of the viral proteins L and NP. The human polymerase I plasmid directs the synthesis of the viral RNA segments. Successful rescue of a recombinant wild-type arenavirus is confirmed by the presence of viral antigens using IFA. At 16 to 18 hours post-infection of Vero cells by tissue culture supernatants, cells are stained with LCMV or candid number 1 specific antibodies. In trisegmented arenavirus rescues, the human polymerase I plasmid is separated into 2 plasmids. In one plasmid, NP is replaced with GFP, and in the other plasmid, the GP is replaced by Gaussian gene. Successful viral rescue can be simply assessed by observing GFP expression by fluorescence microscopy. Furthermore, successful rescue can be confirmed by assessing Gaussian expression.
After watching this video, you should have a good understanding of how to rescue both wild-type and trisegmented arenaviruses in Vero cells with high reproducibility.
We recommend 3 independent transfections for each recombinant virus to increase the likelihood of a successful rescue, as Vero cells are not easily transfected.